drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Elvira Mass, thank you for joining me.
Thank you for inviting me.
Can you tell everyone a little bit about who you are and what you study?
So I'm a professor for developmental immunology at the University of Bonn, and my lab mainly focuses on the macrophage, so part of our innate immune system.
Yeah, and I've had a number of episodes that feature macrophages of different kinds recently. We've talked about them in the context of atherosclerosis, in the context of microglia in the brain, and macrophages are really interesting, it turns out. Some people might know them as merely as immune cells. They go around and eat infected cells and things like that. But what I think a lot of people don't appreciate is how diverse they are and how they develop and change over time, and how there's different macrophages in different parts of the body and different tissues. And I think today we'll talk about macrophages that live or that reside in the liver in particular. But, just talk a little in general terms about like what you study in terms of macrophage biology. Why did you get into this, and why are macrophages as a cell type so important to understand?
Yes, so I'm a developmental biologist by training, and I stumbled a little bit into the immunology field. This was about 2013, and this is when the first studies came out showing that macrophages are actually not coming from the bone marrow, that tissue-resident macrophages are not constantly replenished by monocytes, but that instead we have a yolk sac progenitor. that will give rise to these cells, and that these cells are long-lived and can proliferate and self-maintain without the input of any other progenitor. And this is where I got fascinated to see what these cells are doing in the different tissues, how they are developing. And now we're following this up in my lab, trying to understand what these cells actually need to do during development and tissue homeostasis to really make the tissue work properly.
Mm-hmm. And most tissues have, I guess you would say, their own type of macrophage, or macrophages that become specialized for that tissue. Talk a little bit about that. So like in all of our organs and tissues, there are macrophages that develop for that tissue, meaning that they are somehow becoming specific to how they clean up or how they protect that particular tissue and sort of tuned to a particular organ system.
Yes, exactly. So what I have described in my work as a postdoc is that we have this macrophage precursor, the pre-macrophage, we call it then, that will circulate throughout the whole embryo, and then it will go into every tissue and then become a tissue-specific macrophage once it's differentiates into a macrophage. And then immediately these macrophages are specified. So they take up the niche signals, and then they respond to that and integrate this information into their genome, epigenome, and then also into what they express and do. Of course, every macrophage will phagocytose, so an efferocytose. However, there are special things that these cells need to do, for instance, to provide growth factors or lipoproteins, apolipoproteins, for instance, to neighboring cells for them to function properly. And this can be a metabolically active cell, such as a hepatocyte, or an adipocyte in the white adipose tissue. In the brain, we know microglia. They are important for neurogenesis and neural network function. And this goes on and on like that. And they have specific names. In the liver, they're called Kupffer cells. This is what we really love to study. And yeah, so this is what they do, and we don't really know exactly what they're good for in each and every tissue so far. So it's for development and homeostasis. This is a big black box for many tissues so far.
Yeah. And so I think the important thing to understand up front here is, so there's many different types of macrophages, and they develop. It takes time for them to turn into mature macrophages that are fully functioning and doing what they're supposed to do. Many of them are tissue specific, as we'll discuss for the liver. And my understanding is they can also go bad. So they can probably become malformed and not develop properly. And they can also sort of get sick or go bad. I talked about this in the context of atherosclerosis recently. So-called foam cells are basically sick, or almost you might say obese macrophages. And then they start misbehaving when they become sick and dysfunctional. Does that happen in general in other tissue types? Like, can they sort of go bad?
Yes, generally they can go bad if the environment is going bad. So usually if you have a normally functioning tissue or organ, macrophages will just do their job. And they're very, let's say, tolerant to everything that they see, because otherwise they would be activated all the time. So they eat up a lot of things and they see a lot of things. And this is what I think nature has been taking care of, that they don't over activate.
Yeah.
But if you now go into a high-fat diet or inflammatory situation, then of course the tissue goes bad, and these foamy cells, of course, they are well known. They're also foamy in the brain. Microglia can also become foamy, right? If you have Alzheimer's disease and so on and so forth. And yes, they can go bad. And if they're long-lived, that's a big problem, right? Because they stay there and they're bad there for a long time.
Right. So, by nature, macrophages eat stuff. They clean up debris. They eat infected cells. They're exposed to bad stuff, let's just say. And so I would imagine that, if they overindulge, or if the environment is especially bad, these cells by their nature are being exposed to potentially toxic molecules and things like that, and they themselves can go bad. What is special about the liver macrophages? Do they do anything that is sort of unique that other macrophages aren't doing? What makes them sort of liver specific? What do they have to do in that organ in particular?
Yes, so Kupffer cells, the macrophages of the liver, are specific in that sense because they're one of the very few macrophages that are exposed completely to the blood. So they are sitting within the vasculature, within the sinusoids of the liver, and this is where they sample everything that comes in from the gut. So, and a lot of things are coming in from the gut, fats, lipids, sugar,
right. Everything.
salt, everything.
They're sitting right there, sort of watching and monitoring everything that comes in.
Exactly. And this is why they're so, let's say, important also to keep the liver in a not inflammatory state, to really kind of catch all of this away and not to let it flow through to the rest of the body. And what they're doing in there is not very well understood. Again, during steady state, we are trying now to understand what is happening, for instance, right after birth, because we are kind of cut off from the maternal supply of nutrients. And then we need to really supply ourselves with nutrients. And this is really the switch at birth that we can show that Kupffer cells play a role in controlling the metabolism of newborn hepatocytes. And this is one thing. And then the liver is maturing and a lot of things are happening, like polyploidization. So you increase the DNA content of hepatocytes. That's also not very well understood what this is actually doing and why this is good for. But also here, Kupffer cells seem to play a role.
Wait, so I didn't know. So liver cells are polyploid?
Yes, they are.
Okay, and that for those listening, what does that mean?
It means that you usually have a DNA content that is, we call 2N. That's a normal. Every cell has a certain DNA content, right?
Two copies of every gene.
Exactly. And then the liver, around weaning in mice, but also in humans, this happens during development, it will increase the DNA content. So the nucleus either increases by just growing bigger and become 1 times 4N, or it can become 2 times 2N. So it's like a failure in segregation, let's say, of the DNA after proliferation. But this is done on purpose in certain cells, and hepatocytes are one type of cells where this is happening.
So it sounds like for reasons that are probably somewhat mysterious, hepatocytes, liver cells often have much more DNA in their cells than other the normal cell of the body.
So people think that this is due to, let's say, the toxicity that the liver is exposed to, so kind of a buffering system maybe to what is coming in. But they also say that it could be something that's maybe metabolically related, but it's not quite clear. So causalities have not been shown yet to really say something happens in the polyploidization of the hepatocyte, and then afterwards we have that disease. This has not been really well described.
I see. Okay, so it's still mysterious, but naively I would think, okay, these are liver cells. Liver is our detox organ. Maybe they just have to make twice as many enzymes because they have to turn them over so much or something like that.
Yes. It's very logical, right? But to show it really experimentally is very difficult.
Yeah. So let's turn and talk about pregnancy a little bit before coming back to fatty liver and some of the new work that you've done. So obviously, when a woman gets pregnant, she gets bigger. There's another little body growing inside of her. She has to change, right? The placenta has to be built. Her body just has to change in many ways. Obviously, women get bigger and they put on body weight and body fat when they're pregnant. But what is a normal amount and what is too much, and how has that been changing over time in the modern world?
Yeah, so I think we are changing in the whole world the way that, that pregnant woman or women that want to become pregnant, how they are in terms of their health. Worldwide, right now, we have a number which is really scary, which is around 50% of women being overweight or obese in the age where they can become pregnant. So this has not been an issue at all in the past. Rather, we had more famish. We had too less to eat, especially during wars, for instance, in Russia, but also in Netherlands, very well known, the Dutch hunger winter. So there has been, let's say, there have been studies in the past showing how this maternal health and maternal nutrition is impacting the next generation. And now we're not looking at hunger or underweight, we're looking at overweight, of course. Now, what the mother, of course, is putting on in weight in a normal pregnancy is not, it's normal, right?
Mm-hmm.
This is
Mm-hmm.
like, this will not do anything bad in general. What is bad is if you are already overweight or obese before you become pregnant.
Got it.
That really is the big problem. And we don't really know what this means to the next generation or generations even, because it will have a huge impact on us and how we develop and how we get sick.
Mm-hmm. Mm-hmm. And do we know, like at a correlational level, at epidemiological level, do mothers with pre-existing obesity, when they get pregnant, do they tend to have children that are more prone to obesity and other disorders and diseases?
Yes, this is now being studied more and more. In Europe, there are not super many studies in this direction yet, where we can really follow up big cohorts that are now aging, let's say, because the problem is new, right? So I think the kids that are now being studied are adolescents. In US, I know of different studies. They started earlier, and there I know they're sampling 20, 25-year-old, not children anymore, but coming from obese or overweight mothers. And there are certain correlates, yes. So, of course, obesity in the children born to obese mothers is also logical to some degree, because you are what you eat at the end of the day. However, there is, again, no causality. There is some correlation with neurodevelopmental disorders such as autism, asthma. There are different types of diseases, but again, this needs to be studied in more detail. We need to see also with the aging population how we are going to do. Let's say we in our 20s, 30s, how we're going to be in 20 to 30 years, mm-hmm. coming from obese or lean mothers. This is the comparison we need to do. Mm-hmm. But it has not been that really done so far.
Yeah. And, obesity has become more common over time. What's perhaps not too surprising is, adults, everywhere are more obese now than they have been. What's maybe a little bit more surprising, at least to some people, is that children are also suffering from obesity and metabolic dysfunction at rates, that are unprecedented in history, and from a very, very early age, from a shockingly early age sometimes, or it's almost hard to think about, like, well, how did they become that metabolically dysfunctional that early just from, eating candy or whatever? And you start to think about, well, maybe there was a predisposition here. Maybe they were, maybe before they were even born or conceived, something was preemptively setting them up to be more prone to metabolic dysfunction. I want to talk about fatty liver disease a little bit at a high level. For those that are not familiar, can you just describe what is fatty liver disease and how has it been changing or becoming more common over time in children?
Yeah, so fatty liver disease in general can be segregated into different types. And fatty liver is one of the first steps where we start to store more lipids in hepatocytes. Mm-hmm. And this can progress towards metabolically activated fatty liver, and also to fibrosis, further to cancer. And there is a transition from fatty liver to cancer. Sometimes it goes through a fatty liver and really this metabolic disease. Sometimes it goes directly to fibrosis and cancer. And what I have been told by doctors is that, as you just said, that children are coming into the hospitals and the doctors don't really know what to do, because, and how to change maybe the diet of the children, because they come with really bad livers. And it's not only fatty liver per se, but it can be already worse than that. So yes, this is something that will, I think, become a real problem in the next years and decades.
Yeah, but I think, even just in my lifetime, more or less, like when I was a child, and certainly like before I was born, fatty liver disease used to almost exclusively be called alcoholic fatty liver disease, because you would only see it in middle-aged people that were severe alcoholics, like heavy alcoholics. But now we just call it fatty liver disease because children have it. And so
it's not only children, right? Because it has been dubbed NASH, non-alcoholic steatohepatitis, but now the nomenclature has changed because, as you say, the alcoholism is not really the cause anymore. It's more really the food intake and energy intake that we have, and this is metabolically driven. And yes, children for sure, if they eat a lot of, burgers or drink Coke or other fructose, sucrose, sugar, let's say, drinks, this is a problem per se, but also they have been prepared in utero during gestation while they're in their mother in the womb. They have been prepared already what is to come later. So the mechanisms we don't understand really because we are studying macrophages, but of course also hepatocytes are being programmed by the maternal metabolism. And it is likely that they are responding also differently because these cells are also long-lived, right? So there is an interplay there that we still don't understand quite well, but we will need to be able to maybe help these children and offspring coming from obese mothers.
Yeah, so it's not just that young children are drinking high fructose juice boxes from an early age. They're already set up for that type of environmental stimulus to be even worse than it would have been otherwise based on this kind of developmental programming that happens. And so what we're getting to here is an intergenerational epigenetic effect, which to a naive individual might look like genetic inheritance. But really, we're talking about a developmental effect from the in utero environment, what your mother was doing. And you do inherit that in some sense because your cells get programmed by the environment in utero and what your mom was doing as you're developing, even though it's not a genetic inheritance in the classical sense. Is that roughly how you think about it?
Yes, exactly. So, of course, we can inherit mutations in the genome. This can be bad and can cause different types of diseases. But what we're studying and what you just said is really the epigenome, right? So the epigenetic changes that can then persistently, stay there in the next generation and make cells do something they're not supposed to do. And we try to understand what is the signaling really coming from the mother driving these changes. For now, we try to focus really in the offspring to understand, in our case, what Kupffer cells are doing if they have been exposed to maternal obesity and how they're changing and how this will affect the liver, and whether we can explain really the fact that offspring of obese mothers is developing fatty liver disease, because this is well known. We're not the first ones to show that there is a fatty liver disease after maternal obesity in the offspring. This has been shown in mice and rats, in non-human primates, but even in humans, in human babies, ultrasound studies show that they have lipid accumulation in their liver.
Oh, so we can see this with imaging in humans?
Yes, we can. Yes.
Okay, so even in human beings, you can see the fatty liver forming in utero.
Well, it's not in utero; it's newborn babies.
Newborn. Sorry, yeah.
Yes. So in
but
utero,
already,
it's very early. And then, you just said it like we are preparing the next generation, let's say, to what is to come. Yes, but it's actually interesting because there's this window where the baby is, let's say, more vulnerable to change. So it is in the mice studies that are published has been shown already. So if you switch something directly after birth, it may become worse than you switching diets, for instance, after weaning. So there is like a phase where it will become worse, although you're switching to a more healthy diet.
I see.
And this has been shown already also in this, told before, Dutch hunger winter. So females are starving during the first trimester, for instance, and then they had food again after 3 months. And then 70, 80-year-old people now, they develop cardiovascular diseases, neurodegenerative diseases. This is well correlated by now.
There's critical periods in certain windows of development. And if something happens then, it can lead to a change later on, even if the environment fixes itself later on in development.
Exactly. Because in another hunger, let's say winter in Leningrad, which was much longer, the same happened. The females that were pregnant were, they didn't have anything to eat, but the offspring did not develop that kind of disease as in the Netherlands. So I think this tells you that there is like this window, and you're preparing yourself what is to come. And if it's coming, you can deal with it. You're maybe a little bit overweight, but your liver may be doing much better because, you have prepared yourself to a higher or lower calorie intake. But the switch of the diet, either going too high or too low, then to what you have seen in utero, that is the big problem.
Yeah. Interesting. So there are these windows of development, and I would imagine, right, that a lot of this stuff, when you map it out, it corresponds to the development of things like the Kupffer cells. Like, I'm guessing that there's periods in development, whether it's the first trimester or the last trimester or whatever, you can tell me, when those cells are, being programmed heavily. And if you do something in that window, it's going to have a very different effect on the subsequent child's or adult liver as if you were to do it at a different window of development. So can you talk a little bit about normal Kupffer cell development and programming at a high level? Like when during the pregnancy of a rodent is this programming happening in utero?
So in the mouse, we can see the first Kupffer cell as soon as we have the liver. And this is around E10, so embryonic day 10. A mouse is pregnant for roughly 19 days. But that just because we're kind of stuck with, dissecting a tissue and looking at it. In humans, we know that within the first 4 to 5 weeks, we have the first macrophages developing, and the liver, as soon as it's there, you will have macrophages in there as well. And they, of course, are seeing everything again, what is coming from the mother, because they're exposed to the bloodstream in the beginning. And Kupffer cells, in that sense, are quite interesting because they develop to macrophages. They see their environment. They become tissue specific to the liver. And then they grow together with the organ. And they are within the parenchyma. So they're interacting with hepatocytes, interacting with endothelial cells and other cell types. But then after birth, around 1 week late after birth, you have them migrating to the sinusoids. So they are not always exposed to blood, as we know from adulthood. And this is quite interesting to see how are they programmed, right? Is it directly the maternal blood in the beginning, maybe in the first week or so, or 1.5 weeks? And then it's maybe also what the neighboring cells are giving as information to these Kupffer cells that will have an impact on their programming. In general, this whole journey for this macrophages is necessary for them to function properly. So this all moving around and migrating from one place to the other during development is really necessary. But now if you imagine that you have, short-chain fatty acids or microbiome-derived metabolites from the mother coming in that are not supposed to be there, this will trigger certain programs. And if this then changes the epigenome, so really something that can long-term stay in the cell as information, then this will have an impact on the cellular function and then also tissue function.
So when we think about these Kupffer cells, these resident macrophages of the liver developing, and we say that they're being programmed, should we take that to mean that, okay, the mother's blood is circulating, it's going through the placenta, the baby's organs are seeing the contents of what's in that fluid, it's going to have nutrients, it's going to potentially have toxins, it's going to have all the stuff, much of the stuff that is in the mother's bloodstream, and the particular pattern of nutrients and other things in that blood that the macrophages are exposed to, I would imagine that's going to sort of change which genes are being expressed or not expressed, and therefore change what the macrophages are sensitive to, what they're looking for, how they discriminate between different molecules. Is that roughly what we mean by talking about the programming of these cells?
Yes, absolutely. And I think also the placenta plays a big role there because we don't see everything, right? The macrophage will not see everything that the mother is eating. There are transporters in the placenta that will actively transport certain proteins and metabolites and others not. So I think this we also need to consider because it needs to be strictly controlled. Development needs to be sterile. It needs to be strictly controlled. However, then whatever comes through, and maybe it's rather coming more of something or less of something, that will lead to this kind of programming or reprogramming of macrophages. And yes, they're still developing, so they're still differentiating. It's not like they become suddenly a non-specified macrophage. There's still a Kupffer cell. This we could show experimentally that they are still yolk sac derived. They're not going anywhere. They're still sitting there. However, they are not only responding on a genetic level, let's say, in terms of regulating their genome activity or certain receptors being there and not there. It's also the activity of transcription factors. So usually if you control something via metabolites, you have transcription factors in the cytoplasm of a cell. And then if the cell is sensing something, then it will translocate to the nucleus and then it can become active. And this is something on top of gene expression per se that you can really see in these Kupffer cells happening, that they can really have this molecular switch that is not supposed to be there, at least not at that height, and that this can then reprogram the whole cell long term.
And I would imagine for these cells or any macrophage, it's a really delicate dance, and it's sort of a Goldilocks problem. You want them to be sensitive enough to sense the things they're supposed to detect and get rid of, but not too sensitive, because then maybe you're thinking about autoimmunity and attacking healthy cells. You want them to discriminate between things like at the right level, not too much, not too little. And so this programming is probably pretty delicate and can go wrong in either direction. You can have too much or too little of something, whether it's the expression of a gene or how sensitive they are to, a molecular signature they're supposed to recognize and all that stuff.
Absolutely. I mean, you have so many different receptors on a macrophage surface, and they can bind to the same things, but one is inhibitory and one is activating, right? So, and I think this is kind of what is now just a sponge for a certain molecule to, get rid of the signal and not to overactivate the cell. And how much do I really need to get a signal inside the cell? And this is what I mean, that if we suddenly have something that is usually is a tonus, you can in the background, you can see it flushing through the blood and you sense it a little bit and then it's fine. I just sit there and I don't care of this because I need to take care of something else. But now if the signal is overwhelming and all of your other receptors are maybe full with that signal, then you suddenly signal inside the cell. That's exactly the problem that you have if you overexpose too much with something that may be good in low levels, but they become bad if it's too much.
So let's go through some of the experiments you did in this recent paper. Let's just start at the beginning. So maternal obesity causes fatty liver. What does that look like in your hands? How big is this phenotype? When you say maternal obesity, how are you making the mothers obese and how obese are they getting? And then how bad does the fatty liver look in their offspring?
So the female mice that we feed them with a high-fat diet. It's a lard-based diet. They gain weight. So it's not sucrose or it's just lard. It's just fat.
I want to dig into this because this is important and ties into something I've gotten into in many, many different ways on the podcast. So it's a lard-based diet. So it's high fat and it's pig fat. Can you tell me the actual fatty acid composition of that lard?
Oh, not out of my head, but yes, we have, of course, exactly the atoms, the C16, C18, the palmitate, oleate are the main drivers. But of course, we have a huge list of these fatty acids that are within this diet.
So what I'm interested in is, and some of those listening in the future, I guess, will maybe know what I'm hinting at here. So lard fat I can tell you this is true in the US. Maybe it's not true in Europe, although I suspect it might be. Lards are not ruminants. So their fat basically matches whatever they're fed. And in the United States, pigs are fed soybean and corn-based feeds that are full of polyunsaturated fatty acids. And so lard in the US often has a lot of seed oils in it. And I was just going to ask you if you knew if that was true in this case.
So we are not allowed to import lard-based food for mouse research from the US. So I can tell you that our food is completely different from what you guys have in the US. I am not even sure what our pigs are being fed. Probably very healthy bio something. No, just kidding. It's an interesting question because it's not only the fat, we think, because we're trying now to dig deeper into what is really driving certain phenotypes that we also see outside the liver. And it's probably also the fiber content, because it is changing. Not the diets that we have from the companies that are the control diets are not necessarily the same in terms of fiber. And fat is one component, but I think we're missing much, much more. And what we really would love to do is just to give certain fatty acids to the pregnant mother.
Yeah.
There are ways to do it. You can even follow single fatty acids. It's called click chemistry. So you can really kind of may have a reaction and see it in a mass spectrometer or even by immunofluorescence where the fatty acids go.
But this sounds like this is an important point. So you're inducing obesity with a high-fat diet, and indeed, the diet has higher fat content than the controlled diet. But importantly, the fat content is not all that's different between the experimental and the controlled diet. There's other nutrient differences here.
Yes, absolutely. And it's not only nutrients, because what we have really tried to tackle this down during the revision process to understand what is the upstream signal. We did not put that in the paper because it would have been way too much information. And I think it's going to be a beautiful other paper coming out at some point from the lab. What is changing is the microbiome of the mother. And the microbiome is also producing metabolites. And we do see this microbiome-derived metabolites in the maternal serum, but also in the newborn serum. So it's a mixture of everything. And we were lucky enough to find the downstream signal that is changing. And now we're trying to find basically the upstream signal and the receptors responsible for these kind of reprogramming mechanisms in the embryo.
Okay. Yeah. So give us the basic result here. So how bad is the fatty liver in the offspring from obese mothers?
It is quite okay. If you look at an 11 to 12-week-old mouse, in this case, a male, a female at the same age will not show much. And this is also very well known that females don't develop fatty liver as fast as males. However, if we wait for another 6 to 12 months, almost all of them, including females, will develop a fatty liver, fibrosis, or even liver cancer, although they have never seen high-fat diet in their life. So I think it's a time issue.
So the offspring are never given the high-fat diet. It's just that their mothers were exposed to it before they were even pregnant.
Yes, exactly.
And were they on the high-fat diet during pregnancy as well? I forgot that part. Okay.
They were. You can, and we did these experiments, also not in the paper, but we did these experiments to understand whether the programming can be reversed if we put the mothers on high-fat diet only during pregnancy, or if we put them on control diet and make them lean again and then get them pregnant. So the latter is rescuing the phenotype. So if you become lean, and this is what doctors tell, mothers or to be, you should become lean before you conceive. So this seems to work well. However, there is still sometimes you see fatty liver disease if you just have the diet during pregnancy. So you don't have a metabolic syndrome, you don't have like insulin resistance or anything, but just the diet itself seems to be sufficient to drive partially this fatty liver phenotype.
Okay, and then summarize for me again. So there's a difference between males and females here, and it's not just that the offspring are predisposed to fatty liver disease. You're seeing fibrosis, you're seeing cancer, and other things.
Yes, so this is the, let's say, natural progression of this fatty liver disease. If you don't, stop it, this will progress towards fibrosis. There are certain ways maybe to stop it from fibrosis to cancer, but we always see some animals with really huge liver cancers. And we don't quite understand why this is happening, especially because, like I said before, we have the maternal obesity, we have high fat diet during gestation, but at the day of birth, we put the pups, the offspring, to a controlled diet mother. So we're not changing the mom's diet, right? We're giving the pups to a completely healthy mother. And then this is what happens. And I think this is also something that we would expect to happen in other mammals.
I see. So we shouldn't necessarily think of fatty liver disease and liver fibrosis and liver cancer as completely separate. They can sort of, one basically leads to the other.
Exactly. If you continue to eat unhealthy and, to drink maybe a little bit more alcohol than you should, then it's the normal, let's say this is what I was talking about in NASH, right? The non-alcoholic steatohepatitis, it will go definitely into fibrosis and cancer. And this is very well described and known. And we're trying to work also on how to revert that, because there is sometimes you can see people working on this and trying to find mechanisms to because liver can regenerate quite well, right? It's one of the organs you cut off 90% and it will regrow. So there are ways to make the liver better, but we still don't know the mechanisms or the molecular switches there, what to do with it. But yes, this is definitely also well described in literature, not only for mice, but also for humans, that there is a progression often to carcinoma.
Got it. And so I'm looking here at the diet information. So this is a high fat diet, 60% fat, mostly from lard, 20% carbohydrate. So it's not a ketogenic diet.
No, it's definitely not a ketogenic diet, because ketogenic diet does something completely different, I think, which is also interesting. And we would like to study this more. I'm not sure it's so healthy.
Okay, so let's move on. So you can give these mice fatty liver, and bad stuff can happen if you let them progress. Their mothers are obese on this high fat diet before and during pregnancy, but the offspring themselves are never given the high fat diet. Nonetheless, they have this fatty liver phenotype. What did you guys do next in terms of starting to dig into the developmental programming here of these Kupffer cells?
So the first thing we did is to test the hypothesis whether we do have really persistent changes. We had more groups in that study. We were trying to understand also whether lactational diet would make a difference or not. And it certainly does, but it's not as prominent as maybe in other studies. And this could be due to the type of diet we are using. So we then focused really on the groups of maternal obese and maternal lean mothers, and then having the offspring for most or all of their life on controlled diet. And then we had also diet-induced obesity control group. So after weaning, this is what usually people do. They put adult mice on high-fat diet, and then they study liver disease and metabolic diseases. It was interesting to see that the liver phenotype in terms of lipids and how the liver looked like per se was completely different from a diet-induced obesity. So this maternal obese animals, they had really different types of lipids stored in their liver. And that came along with a change in Kupffer cells and macrophages, especially these signatures were really specific to the maternal diet. So usually you would think what a cell sees at the end of its life, right, when you take it out of the tissue, that this would be the main driver of what you are expressing in terms of genes. However, what we saw is that the Kupffer cells really clustered away from each other, and you could really see that the maternal lean Kupffer cells, independent of the diet at the end of their lives, were clustering all together, and the maternal obese groups clustered completely away, which means really that the maternal diet is controlling what the Kupffer cells will do in their adult life, which was really one of the first experiments where I thought, okay, this is absolutely crazy, but super interesting, and we need to follow this up. And this we then did by looking at the different genes and transcription factors and immunological genes, because we thought, of course, being immunologists, this must be driven by inflammatory signals, something happening in the liver.
So basically the chain of reasoning here is you can give the high fat diet. You can see that the offspring are different when you just look at their livers and overall liver health. You can see that the Kupffer cells are different. And now you're saying, okay, how are they different? We're going to go in and look at individual gene expression and nail down specifically where the difference is coming from.
Exactly. So to understand really what is maybe driving this lipid uptake by hepatocytes, because we know there's paracrine signaling. Kupffer cells will make certain factors, and this has been shown in the context of inflammation, again, like TNF has been shown to drive lipid uptake in hepatocytes. And this was the idea. We thought, okay, you're reprogramming the cells, maybe you're killing them, monocytes are coming in, replacing them, they go crazy, and then the whole tissue goes crazy. Now, we were surprised, but also happy to see that the cells are not replaced. So they stay, they develop at E10.5, and they're still the same cell. So that was an important information. And then we looked into the gene signature, and we saw that there is inflammation. However, we do not think that inflammation is a key driver here of the disease. I think this is something that is happening subsequent to hepatocyte death. Because if they start taking up a lot of lipids, they will die, and then this will drive in other immune cells like neutrophils and monocytes there. So this means that the first step is this reprogramming of Kupffer cells. And then we looked a little bit deeper, and we saw that they're switching their metabolism. So usually they depend on oxidative phosphorylation. And we saw that all of these maternal obese Kupffer cells, that they switched their gene expression to more glycolytic metabolism.
So instead of generating their energy primarily through the mitochondria with oxidative phosphorylation, they're generating their energy primarily through glycolysis. So they're much less efficient at generating ATP?
Exactly. So this is especially in the animals where we had this nutritional mismatch, right? Where you have obesity and high fat diet until your birth, and then you switch them completely. And these animals and Kupffer cells were doing, let's say, worst out of all of the groups. And this is what you can see there. Yes.
Wow. And so that immediately too connects to this whole world, this whole school of thought about how in general, people like Thomas Seyfried, argue that this is exactly what you see, this type of metabolic impairment in cancer cells, generally speaking, that they are impaired in their ability to make energy with oxidative phosphorylation through their mitochondria, and they're glycolytic. They're using this very inefficient means of generating cellular energy.
Yeah, so I think this is what is also known in the field of macrophages as trained immunity or innate immune memory. Of course, this is kind of, this switch from OXPHOS to glycolysis or Warburg effect or whatever you want to call this. And this has been shown that this will change the function of an immune cell, also of a macrophage. Typically, these studies are done on bone marrow-derived macrophages. People have not worked too much really on Kupffer cells per se or in detail within tissues. And we did not really know what it means, right? So what it would mean that the cell is switching its metabolism and how this is done. But then, of course, the whole field knows that this is mainly driven by the transcription factor HIF1 alpha. So upon hypoxia, this transcription factor becomes activated, but it's also known to really swap these metabolic stages of cells. And this is kind of where we're lucky that we got some mice from collaborators to really check this hypothesis. Can we rescue the whole liver phenotype and the whole fatty liver phenotype in the offspring if we take away HIF1 alpha alone? And it worked, right? So if you take out HIF1 alpha and you switch to controlled diet, that is sufficient to rescue the whole liver and also this reprogramming of Kupffer cells.
So a lot of the effect comes from this particular gene program, let's call it.
Yeah, so whatever HIF1 alpha is doing, let's say, it's doing something minor, I would say, until birth, but then a lot of the transcriptional network is changing. So you have one change, but then this will, I think it will amplify. And this is maybe also what is then causing in the long term even cancer, because the cells are getting worse, or they're not getting better at least. And this is something where we see that then HIF-1 alpha in the beginning, but then in other factors such as PPAR alpha or PPAR gamma, also well-known transcription factors important for cellular metabolism, are also completely changed. So this is something that we try to understand in more detail and depth, what is actually happening later on, but we really don't know for sure.
Yeah. And this is maybe a sort of a general thing that people should understand is, sometimes you have, what you might call a master regulator of transcription. So like one gene might turn on at some point in development, but that gene could be a transcription factor that turns on 10 or 20 or 50 other genes. And so even though it's, only one gene, it's this critical regulator that's going to have a bunch of other things changing after it.
Yeah, exactly. And this is true for HIF-1 alpha for sure. So this transcription factor, this gene really controls many, many genes. Not only, as I said, metabolism, but this is actually was described for the first time to respond to hypoxic conditions, right? Too less oxygen. Has nothing to do with metabolism per se, but nevertheless, it's one key regulator in many processes, and it seems to be the case also for this maternal obesity or whatever it is being sensed by, right? So whatever it sees upstream, this we don't know yet. But it will turn out that HIF-1 alpha will go more to the nucleus and will start being active and upregulate genes at birth. Yeah.
And were you guys able to just explicitly connect the dots between these reprogrammed Kupffer cells, what they're doing differently, and the fatty liver phenotype per se? Like, did you figure out exactly how the Kupffer cells in mice that had obese mothers, how are their Kupffer cells behaving differently, and how is that actually resulting in lipid accumulation at a higher rate?
Yeah, so we really digged very deep into the mechanism, and we saw that there were certain factors that these Kupffer cells suddenly started to produce in general, or to produce more of them. As I said, it's okay and it's required to have certain things to be produced, growth factors, apolipoproteins, you need them. But if you make too much of them, then the neighboring cell, in this case, the hepatocyte, will not be happy. And this is exactly what is happening. So we saw on a transcriptional level that this whole reprogramming leads to the upregulation of apolipoproteins, such as ApoA and ApoE. And that this then is sufficient if you give these lipoproteins to hepatocytes, that this is sufficient for them to take up more lipids. So it seems to be really a driver of lipid uptake in hepatocytes in general. And of course, if you have this neighboring, this neighborhood between the cells, you can assume that this is going to be then the crosstalk at the end. It's for sure paracrine signaling. This we figured out with very sophisticated, I would say, ex vivo assays where you kind of put hepatocytes on the bottom, Kupffer cells on the top, and you have a lot of medium in between, so you can really exchange molecules from one cell to the other. So we know it needs to be Kupffer cell dependent, and it's apolipoproteins, but I doubt that this is the only thing that's happening.
Yeah. And so, I mean, I'll just ask it this way. Why do you think these Kupffer cells are becoming glycolytic? Like what, like from their perspective, if we anthropomorphize, what exactly is pushing them to be glycolytic, and why can't they use oxidative phosphorylation?
I think this comes all back to this kind of idea of preparing yourself what's to come. So you're in utero and you see all of this metabolites and lipids, and then you expect it to be the same if you're born.
Yeah.
And I think this is something that you need to be prepared for, right? You will have excess energy anyway. So, you are probably busy doing something else in that sense, because also glucose levels are probably very high. Macrophages have also glucose transporters, right? So everything is changing and ready to go to be kind of obese, and suddenly you're changing the diet. And I think this is the overwhelming situation there that they really cannot handle. They cannot handle this different type of diet and this different type of intake of metabolites. And why exactly they go into glycolysis? I mean, it's not like it's completely shut down or, Oxford, there's still something there. It's somewhere in between.
They're just shifted. They're Warburg shifted.
Exactly.
Yeah. Interesting. So, one way to think about all this and tie it back to those old studies in humans when there were famines and stuff is it's as if, right, everything's trying to be set up to match what was happening in the world when you were in utero. So if you're in a time of famine, it's like your body's being prepared to be in famine conditions. And if it's the opposite, your body's preparing for the opposite. So then when there's a mismatch between what was prepared based on what your in utero environment was like and what your mother's environment was like with the environment you're then born into and living in, the mismatch is what causes an issue here. But it's almost like development is almost smartly trying to prepare you for what your mom was experiencing.
Yeah, exactly. And I think it's also true not only for macrophages, but also for metabolically active cells, because hepatocytes, they also react to metabolites, and if they react in a certain way to one metabolite, and if it's too much, at some point they shut down certain programs because you cannot react all the time, right? You cannot keep on taking on and on and on. And I think this is true for any metabolically active cell or long-lived cell, right? We just were lucky enough to kind of pinpoint a certain thing to macrophages. But I think there is much more happening, because what we kind of went over in the paper, because it was not relevant at that point, because we were focusing on this nutritional mismatch. Now, if you keep the animals on the high-fat diet, so the offspring, and you are HIF1 alpha knockout, you don't rescue anymore. So you still develop a fatty liver disease. So it is not sufficient to knock out HIF1 alpha postnatally. It is sufficient in this developmental time window of gestation. And if you do the switch, then you rescue. So this is important during embryogenesis in the womb at the end of the day. But then if you're born and you stay on high-fat diet, of course, you also will develop a fatty liver. But then you don't rescue if you take out HIF1 alpha. So something must come on top. Once you're out of, you're out and you take up milk, you eat, then something else is happening on top. And this we don't know yet either, what is happening there. So there are many factors. And we found the one in this embryonic developmental window that seems to play a role and really control long-term liver homeostasis.
Yeah. But I mean, a lot of these results and other things imply that it's really important to, if you were a woman and you were, planning to be pregnant, it's really important to think about the pregnancy the whole time, start to finish, because there are things that have been observed where, if something bad or wrong happens, let's say early in development, even if you fix your diet or whatever later on, it can be too late in some sense.
Exactly. It already starts at the oocyte, right?
Yeah.
So this has also been shown that, of course, metabolic syndrome, and also type 2 diabetes, etc., it plays a role into how your oocytes are in terms of health and what does it mean to the next generation. In our case, we could pinpoint this by our genetic models really to the macrophage. But yes, for sure, it's important to really be healthy throughout the whole pregnancy, because you never know, immune cells develop super early. And what we also see in our studies, that only macrophages are changed, that it's likely also the hematopoietic stem cells that are changed. So you can rescue the whole thing if you take out this bad macrophages and you give it naive precursors back, monocytes back, they make new Kupffer cells and they can rescue. But if you kill them and you give them back their own, progenitors, so they have been probably also reprogrammed, because they don't rescue. So whatever is in the bone marrow giving rise to all the different blood cells is likely also altered for life. So we're just kind of, at the tip of the iceberg, I think, now with this liver story, because this is what is directly visible if you open up the animal. And what has been described in humans, but I think there's many, many more phenotypes that we need to still look at.
Yeah. And so, if we connect the dots here and we extrapolate to the extent that results like this extrapolate to humans, which they probably do to some extent, it starts to make sense for why we're seeing so much fatty liver disease so early in even very, very young children. It's not just that they're drinking juice boxes and soda from an early age. That's a factor, but they're already predisposed for that stuff to be even worse for their liver health because of these kind of epigenetic changes that are happening from not just from the in utero environment of their mother, but probably from the very moment they were conceived.
Yeah, exactly. And we were speaking now just about the mother, but of course the father also plays a
Yeah,
role.
yeah.
So, there is no causality shown yet, but for sure, F1 and F2 generations, and I think it's daughters in mouse and rat studies, where it has been shown that if the father is obese, that this will also lead to metabolic problems in females in the F1, F2 generation. So there are so many factors that one would need to consider. And of course, everything comes together. We are working with mice here. There are, specific pathogen-free mice. They're very defined. They're inbred strains. We are, of
These are
course, not
pathogen-free mice that you're working with?
specific pathogen-free, so SPF mice.
Oh,
They are quite defined in terms of the microbiome. And this is why I think it is working all out like that. But for us humans, we need to consider that we are exposed to different things and different microbiomes. We are getting sick in between. Mice don't get sick, right? They're kept super healthy and all the time because this is what scientists need to do to have reproducible results.
yeah, yeah.
And this is where I think we're kind of like in the worst situation, or we end up in the worst situation with these mice. Doesn't mean that it has to be like that in humans, but likely it is like that because, as you say, children coming in and they don't even have to eat unhealthy or drink
Right.
anything. I think even if they eat healthy and, maybe the parents did switch the diet, but this may be even worse for the liver, at least, right? They will be
Yeah,
lean, but the liver is going to
yeah.
be a problem.
Essentially, what you're saying is your results in rodents imply that if the rodent is born to a mother that was obese because of using a high fat diet, its liver is basically being programmed to expect a high fat diet. And then if you give it a non high fat diet, something that's quote unquote healthier, that can actually be more problematic because now you're not giving it a diet that it was preparing for.
Exactly. So I think this is why it is important. Whatever you do, you just keep doing for a while until this kind of window is
Yeah.
closing. And then you can maybe start adjusting diets also for a child, right? I mean, child should not be on a diet in general, I
Yeah,
think.
yeah, yeah. But I mean, is that maybe a heuristic that you take away from this and related work, which is that, like how would you start to think about pregnancy if you were going to get pregnant or something? Like, how do you walk the line between not too much, not too little? Would you recommend fasting for a pregnant woman, or would you just recommend, hey, no matter what you do, it should be consistent throughout the pregnancy? Like, how do you start to think about those parameters?
Right. So I think consistency is very important. I think if you are maybe eating a little bit unhealthy, you should change it way before you are planning a child, because you have it in your own hands to really reprogram back your body and
Mm-hmm.
maybe also your microbiome, which will play a role. But then changing everything is also not good because you kind of adjust it to your life and what you do and what you eat. Typically, if you change everything, that will also start reprogramming your own body,
Mm-hmm.
and that may or may not be good in this moment. I would not fast. I think fasting is I mean, if you're fasting anyway all the time and you do intermittent fasting all your life, probably that's a good way to continue this, but please speak to a doctor beforehand. um, I think you will need to change a lot of things before you are pregnant. During pregnancy, I think it's a really bad thing. And
Mm-hmm.
also directly after birth also would be a bad thing.
Mm-hmm.
It's better to have maybe a little bit overweight than having really kind of a liver disease progression.
Yeah, yeah, yeah. But basically what's implied here is that the baby's body and the baby's liver right after birth is more or less going to be expecting the same type of
Yeah,
stuff to be
exactly.
coming in
Yeah,
as it was when it was inside the mother.
exactly. Yeah.
Interesting. What are some of the next steps here in terms of the follow-up work that you guys are doing?
Yeah, so I know you talked to Justin Perry recently, and
Yes.
we also are in touch with him because we're also working on microglia in this context. We're trying really to understand how the different tissue-specific macrophages are programmed by maternal obesity differently. That's one big question. And then we're also trying to understand beyond obesity, what other type of environmental factors will play a role in programming macrophages. And here we focus mostly on microplastics because similar to metabolites or other factors that macrophages express maybe receptors for can sense, microplastics will be eaten up by macrophages, and they will eat and eat and eat, and they will become dysfunctional, and then the tissue is also not healthy anymore. So that's the idea we're following up now, to really understand how the whole environment, because obesity is one thing, and then maybe microplastic and other toxins another, and then we need to combine it all together, I think.
Okay, interesting. And I would love to know more about these diets. I feel like I need to start doing this on the podcast more. The whole diet literature has a lot of problems in my view. A lot of these diets, we don't actually know what's in them. To the extent the manufacturer even knows, sometimes they're wrong. People who have really dug into this have found that they're even wrong. I would love if at some point you and other people using high fat diets like this can go in and really look at the fatty acid composition in particular of these diets. And at some point, I mean, this is almost an absurd request. I'm not expecting you to do this, but somebody needs to actually independently verify what these diets are, because everybody's taking the manufacturers at their words. And I suspect if someone does that study, if someone actually independently takes the cube of mouse chow and does a nutritional profile on it, I'm not going to be surprised if it doesn't really match what the manufacturer puts on the label. I think a lot of these high fat diets are, we know for a fact they don't actually model the fatty acid profile that a Western human is consuming today. And I think a lot of these high fat diets have a high PUFA load and probably a higher PUFA load than what we think they do.
You're absolutely right. Yeah. I think so. We do have a biochemist in house, and we have like a big consortium on metaflammation at the University of Bonn. So we are really taking care that we feeding the animals what we actually think we're feeding them. So this has been tested. People have tested this, and this is super interesting what you're saying. And this is where I have always discussions with different scientists where they come up to me and say, hey, how do you get your like females obese at all? Mm-hmm. This never works for me. Or how do you get this phenotype? And the response I always have to give them, or what I ask them, is what type of controls do you have? And they say, yeah, chow diet. I'm like, yeah, but can you explain to me what chow diet is?
Right,
and this is exactly what you're saying, because this is yeast, and this is depending on the weather last year. This will have completely different components. And this is what people don't take into account. They just order the high-fat diet from the company and then compare it to their in-house chow diet. Right. And this is usually a high-fat diet.
Right. And it's like already high fat, and they don't actually know the fatty acid profile. From head-to-head comparisons I've seen in the literature, the best diet at making mice fat is high in linoleic acid. So there's lots of different high fat diets, but even better than like a 40% fructose diet, the high linoleic acid high fat diet, from the data I've seen, is the best diet at making a mouse fat.
Yeah, and this is something, again, I think you go towards what we would love to do, to test single fatty acids and give it to the mother during pregnancy to see what is really key, or maybe a combination thereof, right? So we need to understand this, and yeah, we should write this review.
Yeah. Well, yeah, I mean, absolutely. And there's another paper I wrote about, I can't remember the author off the top of my head, but the essence of the paper was that they were looking at things like high fat diets, and, diet 1, diet 2 have different effects. And you would think, I think one of them was a ketogenic diet. You would think, oh, well, the most salient difference between diet 1 and diet 2 was its fat content. And they tested it, and it turned out it had nothing to do with macronutrients, the phenotype they were looking at. It had to do with the absence of a plant-derived phytoestrogen that the microbiome was metabolizing into some other, like it had nothing to do with macronutrients at all, even though everyone would have thought so ahead of time. So these diets are different in many ways, and we often don't realize how all the ways that they're different and which one's important.
Absolutely. And we have a very interesting phenotype where we can rescue the death of a mouse, a genetic knockout, by giving the mom high-fat diet and the offspring as well. Now, if we put the offspring to this nutritional mismatch from high-fat to controlled diet, they die. We don't know why. So we don't know what's missing in the controlled diet in this case.
Right.
So exactly into this direction, this really drives me a little bit crazy, because usually people don't think about this, right? They just give diet 8 weeks, 16 weeks, and then you have a phenotype. I think we need to really consider this and mix as well. Have cholesterol, sugar, salt. We're always forgetting the salt because we're working on a high salt diet, and it's never within the high fat diets, because if we do eat unhealthy, we typically take in a lot of salt as well, and it's also doing something to the different tissues.
Is there anything you want to reiterate, or any final thoughts you want to leave people with about anything that we discussed today?
Well, I think we should think about what we eat, but also maybe have in mind that we not always control our health and how healthy we can be and become, because maybe a lot of things have been preset by our parents and we don't know it yet. But I think we as scientists are really working towards understanding the mechanisms and also trying to help revert these programming effects if they're bad. So this is something where we're trying to work towards too with many great colleagues, and looking forward to the next decades of science into that direction.
All right. Well, Professor Elvira Mass, thank you for joining me all the way from Germany, and I look forward to talking to you again in the future.
Thanks so much.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about one of my cool new affiliate partners called Seed Oil Scout. Seed Oil Scout has a really cool map feature that helps you find restaurants in your area where you can find seed oil-free options. They also have a really cool product scanner, so you can take out the app and use the camera functionality of your phone to scan products in grocery stores and see what's actually in these things. Do they contain seed oils? Do they contain other things that you want to know about that aren't necessarily on the box? This will help you find places to eat and find foods to eat that actually have what you want in them and help you avoid those that don't. So if you're looking to cut back on your seed oil consumption, or you just want to know more about what's in the foods on the shelves at the grocery stores, download Seed Oil Scout today. If you go through the link in the episode description, click into that and you'll see a link to the Seed Oil Scout app. If you go through that link on my support page, you'll be directly helping the podcast, and you'll be able to download this really cool, easy to use app that will help you find healthier food options. So go through the link in the episode description and download Seed Oil Scout today.